Today in this video we are going to study about the structure of DNA. Structure of DNA. DNA stands for deoxyribonucleic acid. DNA stands for deoxyribonucleic acid and it is the genetic material. DNA is the genetic material and the main function of DNA is to store and code genetic informations. The main function of DNA is to store and code genetic information and the DNA is located inside the nucleus. So DNA is the genetic material and the main function is to store and code genetic information and DNA stands for deoxyribonucleic acids and it is located inside the nucleus. Now we are going to study about the double helix model of DNA. The structure of DNA, it is double helix model of DNA. The double helix model of DNA was discovered by Francis Crick, James Watson, Maurice Wilkins and Rosalind Franklin. The double helical structure of DNA was discovered by Francis Crick, James Watson, Maurice Wilkinson and Rosalind Franklin. DNA is a polymer of nucleotides. DNA is a polymer of nucleotides. What is meant by polymer? Polymers are repeated units of monomers. Polymers are repeated units of monomers. Here in the case of DNA, the monomers are nucleotides. Monomers are nucleotides. So DNA is a polymer of nucleotides. Polymers means they are repeated units of monomers. In the case of DNA, nucleotides are the monomers. Then what is meant by a nucleotide? A nucleotide consists of three components. They are a phosphate group, a sugar group and the nitrogenous base. Phosphate group and sugar group and nitrogenous base. The sugar groups are deoxyribose or ribose. Sugar means it is a pentose sugar and that is deoxyribose or ribose. And the nitrogenous bases are adenine, thymine, guanine, cytosine. Adenine, thymine, guanine, cytosine. These are organic molecules. So the nucleotide consists of three components. They are phosphate, sugar and nitrogenous bases. The sugar may be deoxyribose or ribose sugar. It is a pentose sugar. And the nitrogen, there are four different types of nitrogenous bases. They are adenine, thymine, guanine, cytosine. They are organic molecules. These are the sugars, pentose sugar, ribose, which is present in the nucleotides. One is deoxyribose and the other one is ribose. The only difference is in this second carbon. The only difference between these two is in the second carbon. Here the H, OH, H and H, and in the case of this H and OH in the second place, second position. So this is the ribose sugar. It is a pentose sugar. That is 1, 2, 3, 4, 5. It is a 5 carbon sugar. So it is a pentose sugar and two different types of riboses are there in the nucleotides. They are deoxyribose and ribose. And the only difference between these two is in the second carbon. This is the phosphate group present in the nucleotide. So one phosphate ion attached to four different oxygen atoms. This is the phosphate group in the nucleotides. Now coming to the nitrogenous bases. There are two types of nitrogenous bases. They are purines and pyrimidines. Purines and pyrimidines. Purines are two types. They are adenine and guanine. They are double ringed structures. Purines have double ringed structures. And pyrimidines are cytosine and thymine. They are single ringed structures. Pyrimidines are single ringed structures. This is the structure of adenine, then guanine, cytosine and thymine. These are the nitrogenous bases present in the DNA. or in a nucleotide. These are organic molecules and are basic in nature. Now we got an idea about the structure of a nucleotide present in the DNA. So it consists of a sugar, pentose sugar, that is deoxyribose. In the case of RNA, it is ribose. And nitrogenous bases, they are adenine, cytosine, guanine, thymine. Adenine, cytosine, guanine, thymine. In the case of RNA, it is adenine, cytosine, guanine and uracil. Adenine, cytosine, guanine and uracil. And a phosphate group is present at the 5' end of the ribose, deoxyribose. So this is the structure of a nucleotide. Now DNA is a polymer of nucleotides. So these monomeric units are linked together to form the DNA. These monomeric units of nucleotides are linked together to form DNA. Let us see the structure. So these are the nucleotides. This is one nucleotide, second, third and fourth nucleotides. DNA is a polymer of nucleotides, and we know that this is the first carbon, second, third, fourth and fifth. Okay, so fifth carbon, CH2OH. So DNA is formed by 5' 3' phosphodiester bond. Phosphodiester bond. DNA is formed by 5' 3' phosphodiester bond. So here we can see this is the fifth position, 5' position. So 5' to 3'. Again 5' to 3'. This also like this. So in the case of DNA, a sugar phosphate backbone is there, to which nitrogenous bases are attached. Sugar phosphate backbone to which the nitrogenous bases are attached in the case of DNA. So these are the nucleotides. And these nucleotides are polymerizes to form DNA. This is one nucleotide, this is the second one, then third and fourth. Here you can see 4 nucleotides, and they are linked by the nitrogenous bases, linked by sugar phosphate backbone. They are 5' 3' phosphodiester linkage. This is the 5' position, this is the 3' position. So the linkage is like between these two. Here also like this. So we get a structure like this. So these are the nitrogenous bases. This is purines, pyrimidines. This is another pyrimidine. This is also another pyrimidine. Purines are double ringed and pyrimidines are single ringed. Purines are double ringed and pyrimidines are single ringed. So these are the structure of nucleotides, and these nucleotides are joined together by phosphodiester bond to form the DNA. We all know that DNA is double-stranded. So we can see a strand similar to this in the opposite side also. This is one strand and this is another strand. These strands are anti-parallel DNA strands, because here the 3' end, here the 5' end. In the case of here, this 3' end, this is 5' end. So these two are running in different directions. These two strands are running in different directions. First one is 3' to 5' direction. The other one is in this direction. This is in this direction. So this is anti-parallel DNA strands. They are anti-parallel DNA strands. If we twist we get double stranded DNA, double helical DNA strand. If it twist, we will get double helical DNA strand. Here, inside the strand, the nitrogenous bases are hydrogen bonded. That is, cytosine always pairs with the guanine and adenine always pairs with the thymine. So, adenine pairs with the thymine with the 2 hydrogen bonds. Similarly, cytosine pairs with the guanine with 3 hydrogen bonds. 3 hydrogen bonds. So, in the DNA, we can see a sugar phosphate backbone and to which nitrogenous basis is attached. Similar type of strand is there in the other side, and both they are hydrogen bonded between the nitrogenous bases. This is the structure of DNA. Now we can see the helical structure. This is the double helix structure of DNA. Here you can see sugar phosphate backbone. This is the sugar phosphate backbone, and 2 anti-parallel strands are joined by the hydrogen bonds between the nitrogenous bases. Hydrogen bonds, these are the nitrogenous bases. They attach together by the hydrogen bonds. Guanine pairs with the cytosine with the 3 hydrogen bonds. Similarly, thymine pairs with adenine with 2 hydrogen bonds. So, this is the structure of DNA, double helical structure of DNA, and it has 2 grooves. This is minor groove. This is minor groove, and this is major groove. Major groove. 2 grooves. They are minor grooves and major grooves. In the double helix of DNA, sugar phosphate backbones are there. Sugar phosphate backbone and 2 strands, anti-parallel strands, are joined by the hydrogen bonds. These are the hydrogen bonds. The hydrogen bonds are between the nitrogenous bases in each strand. So, the adenine always pairs with the thymine, guanine pairs with the cytosine. The diameter of the DNA is 2 nanometer. The diameter is 2 nanometer. Then, here you can see the minor groove. This is the major groove. This is the major groove and this is a minor groove. So, you can see alternate major and minor grooves in the double helical structure of DNA, and the length of one turn is 3.4 nanometer. Length of one turn contain is 3.4 nanometer. Length of one turn is 3.4 nanometer, and each turn, one turn contain around 10 base pairs. One turn contain around 10 base pairs. So, the distance between 2 adjacent base pairs, the distance between 2 here and here, the distance between 2 adjacent base pairs is 0.34 nanometer. 0.34 nanometer. Even though the nitrogenous bases are basic in nature, the DNA double helix is acidic because of the presence of phosphate groups, phosphoric acid or phosphate groups. If we untwist this DNA, untwist this DNA, we get a DNA ladder like this, DNA ladder. Here this is the sugar phosphate backbone. These are the nitrogenous bases. They will share the hydrogen bonds. Okay. Now let us summarize the structure of double helical structure of DNA. DNA, it is a polymer of nucleotides. It is a polymer of nucleotides. A nucleotide consists of phosphate, sugar deoxyribose or ribose, and nitrogenous bases. Nitrogenous bases are adenine, thymine, guanine, cytosine. These are the structures of deoxyribose and ribose. The only difference is in the second carbon atom. This is the phosphate group present in the nucleotide. These are the nitrogenous bases. There are two types of nitrogenous bases, purines and pyrimidines. Two types of pyrimidines are adenine and guanine. They are double ringed structures, and pyrimidines are cytosine and thymine. They have single ring structure. This is the structure of a DNA nucleotide. Here you can see a pentose sugar and the nitrogenous bases and a phosphate group. This is a single nucleotide. And the nucleotides are joined in a complex manner to form the double helical structure of DNA. Here the 5' phosphate, and here this is a 3' hydroxyl group. So the phosphodiester bond is in between 5' phosphate and 3' hydroxyl group of the pentose sugar. So the bond is 5' 3' phosphodiester bond. We all know that DNA is double helix structure. So it consists of two anti-parallel strands. One is in the 3' 5' direction, other is in this 3' 5' direction. So two anti-parallel strands, they are joined together by the hydrogen bond between the nitrogenous bases. Here adenine always pairs with the thymine and guanine always pairs with the cytosine. Guanine pairs with the cytosine with the 3 hydrogen bonds, but adenine pairs with the thymine with the 2 hydrogen bonds. This is the double helix structure of DNA. Here you can see the sugar phosphate backbone and the nitrogenous bases are inside. They bonded together with the hydrogen bonds. The diameter of the DNA is 2 nanometer and it has minor grooves and major grooves, alternate minor and major grooves, and around one turn contain 10 base pairs, and length of one turn is 3.4 nanometer. Length of one turn is 3.4 nanometer, and the distance between two adjacent base pairs are these two. The distance between two adjacent base pairs are 0.34 nanometer. Even though the nitrogenous bases are basic in nature, the DNA is acidic because of the presence of the phosphate group. This is about the structure of DNA. Thank you.